Hello judges, I'm going to present to you my project today, Synthera, a probiotic swarm for therapy for cancer. Okay, the problem. Cancer is the number 2 cause of death worldwide, which accounts for 10 million people who die a year. That's 1 out of 6 deaths. Current treatments are limited by their intense side effects and cost. Many patients also relapse because their therapies don't address tumor microenvironments. Tumor microenvironment challenge. Tumors are heterogeneous ecosystems, which means they have hypoxia, high lactate and acidic pH, immune suppression zones. Conventional drugs struggle to selectively penetrate tumors. The need is living therapies that adapt inside tumors. Our solution, Synthera, engineered probiotics programmed to sense tumor conditions. They are encapsulated in oral capsules, which make sure that it is safe delivery all the way to the tumor. So the mechanisms. First, we detect tumor hypoxia and acidity. Then we swarm inside the tumor tissue. On the third step, we have 2 different options depending on the different type of patients. If our patient has a high immune system, then we will activate the immune activators, which summon the CAR T cells to destroy the tumor. If our patient has a low immune system, we'll use oncotoxin to directly kill the tumor ourselves. Okay, and last step for all of that is to self-destruct via kill switches so that the bacteria will not overpopulate and circulate inside your body. Composition and mechanisms. Our capsule design is made with a biodegradable PLJ shell, which means that it can have a timed release. Our probiotic core, which is engineered E. coli Nystal 1917. Tumor sensors detect hypoxia and high lactic. And we have a triple kill switch for safety. So our payloads for the oncology is oncolytic peptides, which kill tumor cells, cytokines that can activate immune cells, and anti-angiogenesis proteins, which cut off blood supply to the tumor. Okay, our demo. So this is my demo that I coded for our capsule. Okay, oncology simulation demo. So first, the capsule enters the bloodstream. Then probiotics is released near the tumor. Swarm formation, payload release, tumor shrinks, and bacteria self-destructs. Mission accomplished. Manufacturing workflow. So we have steps on manufacturing it. First, we do the strain engineering, CRISPR tumor sensing circuits inside the bacteria. We have fermentation, GMP compliant bioreactors to make that bacteria. Encapsulation, FDA approved PLJ shell, quality control, DNA sequencing and biosafety checks, and formulation, which creates self-stable capsules. Feasibility. Our prototype cost per 100 capsules is less than $1,200, which we can scale down to less than $10 per capsule versus the $20,000 to $100,000 standard oncology treatments per month. Okay, comparison with traditional therapies. So, Synthera wins with all of these criteria and features. First, we have high tumor specificity. Then we have low toxicity for our good cells. Our cost is the lowest by many times, and our oral capsule features delivery that you can do anywhere in the world at home without having to find, for example, an IV clinic, radiation lab, or infusion clinics, which might not be existent in your current county or city. And finally, our relapse prevention is strong because we address the tumor microenvironments. Okay, market opportunity. The oncology market now is over $200 billion annually. Precision therapies are the fastest growing segment. Synthera disrupts with low cost and living capsules and enables decentralized local production worldwide, so everyone in the world can access this treatment. Safety and oncology use. Our safety is based on the grass probiotic strain, which is proven to work. Our triple kill switch system prevents uncontrolled growth. Biodegradable capsules ensure controlled release and designed to meet oncology biologic safety standards. Our next steps. Our next steps will include building a prototype in academic labs, validating in tumor-originated models, examine in animal xenograft studies, partner with oncology biotech accelerators, and move toward clinical trial pathway. Closing. Okay, vote for Synthera, redefining cancer therapy. It is safe, targeted, affordable, scalable, and can be used anywhere in the world. We are turning probiotics into a living weapon against cancer. Okay, here's my contact information, which is my Gmail. Here's the link to our demo, which I will show you in a second. And here's the QR code to the demo. Okay, I will now take you a short tour around my demo website. Okay, so our topic is about engineering probiotic therapeutics. Okay, precision tumor targeting bacterial platform for cancer therapy. Here is our data on molecular engineering, target mechanisms, safety systems, clinical data, prevention, and future. Okay, let's look through the interfaces of the research. Okay, first we have the delivery vehicle. So this is our capsule. Okay, these are our parameters, and here's an intro on our capsule. Biodegradable pharmaceutical capsule engineered for targeted intestinal release and bacterial payload protection. Okay. Here are our bacteria. So the E. coli Nersal 1917 strain genetically modified with tumor sensing circuits and therapeutic payload genes. So here are our genetic components, functions, and copy number. Our biosensor array to detect our tumor target and not release the bacteria while having a false alarm. So like this prevents us from releasing the bacteria at our tissues or our good cells. So the 6 parameters are designed to detect the unique physiological signature of solid tumors. Our logic gated activation. If 5 over 6 are active of the sensors are active out of this array, we will have the therapeutic activation on. If the output if the 1/6 of the sensors are active, the therapeutic activation is off. So this is our logic gate for the parameters for payload of bacteria. Okay, the technical specification. This is our performance metrics and our molecular specifications. You can see that our detection sensitivity and specificity and false positive rate are the lowest. Pharmacokinetics. This is how it works, like the whole train of deployment, along with the times and methods and parameters of release. Here are our data, simulation data against cancer cells. So our method proves very effective in most sorts of cancers. Okay, our safety profile, we have a 3 triple kill biocontaminant architecture that makes a triple kill switch. Our primary kill switch is synthetic leucine oxytrophy, prevents survival without external supplementation. Secondary kill switch, temperature-sensitive toxin activated above 42 degrees Celsius threshold, temporal controlled, programmed senescence ensures the maximum 96-hour lifespan, which includes enough time to kill the tumor and exit. This is our data. Okay, this is our simulation also shown in the slides. So here we have the ingestion of the capsule, the tumor target accumulation, biosensor activation loggage gate, tumors here, the therapeutic payload released, which attacks the tumor and kills it, tumor destruction, target is gone, and self-destruct. Okay. We have our cancer prevention protocol. So, our prophylactic cancer prevention protocol works with a 97% prevention efficacy and 99.1 early detection sensitivity. This makes sure that if you were to be diagnosed with a cancer and use our method, and the cancer dies, the patient will not relapse from the cancer again. Okay, here's our data from the tests. Future applications. So next, our future applications are neurological, cardiovascular, and infectious diseases. So our global health impact projections. Lives saved annually, 12.4 million people saved by 2035. Economic impact, $2.8 trillion healthcare cost reduction, which is a huge amount of money saved by our program. Here are our data during stages. Okay, here methodology. We use our strain engineering, biosensor integration, logic key implementation, and safety system design. Okay. Thank you all judges for listening to me today. Remember to vote for Synthera to save more than 1 out of 6 people that die every year, and to make sure that cancer therapy is available for everyone all around the world. Thank you, judges. Bye.